Okay, let's unpack this. Measles. You know that childhood illness we mostly hear about as a thing of the past?
Hmm.
Or maybe occasionally in the news a local outbreak?
Right. Seems almost historical sometimes.
Exactly. But what if I told you there's this entire unseen world beneath that familiar rash? A world of, like, molecular detective work that's absolutely crucial to understanding, tracking, and, potentially eradicating this disease.
It's fascinating stuff, really, deep down at the genetic level.
So today we're taking a deep dive into that microscopic universe of the measles virus. We'll look at its building blocks, how it spreads, and the cutting-edge scientists use to track it globally.
Yeah, we've pulled together insights from a bunch of scientific papers, WHO guidelines, the latest research.
Trying to give you a shortcut, really.
Mm.
Our mission is just to pull out the most important nuggets so you can walk away with some surprising facts, maybe a few aha moments about this persistent viral foe.
Sounds good. Where should we start?
So let's begin with the absolute basics. For someone who might only vaguely remember measles from, I don't know, health class,
Hmm.
what is it exactly? And what does it actually look like when someone gets sick?
Okay. So measles is an incredibly contagious illness. It starts with fever, a cough, a runny nose. Doctors call that coryza,
Right, like a really bad cold to start.
pretty much, and red, watery eyes, conjunctivitis. Then typically you get that characteristic red, blotchy rash. It's called a maculopapular rash. Spreads all over.
Okay, and it's caused by the measles virus. Obviously, you mentioned it's a specific type.
Yeah, it's a single-stranded, non-segmented, negative-sense RNA virus. Bit of a mouthful.
Whoa, okay. Negative sense RNA virus. What does negative sense actually mean, like practically, for how the virus works inside our cells?
That's a great question. Think of it like a photonegative. Our cells can't directly read the genetic info on that negative strand to make new viral proteins.
Oh.
The virus first has to make a positive copy, kind of like developing the photo.
Mm-hmm.
That positive copy is what our cell machinery can read. It's an extra step it has to take.
Got it. And you mentioned it belongs to a family, the morbillivirus group. Sounds like it has relatives.
It does indeed. Other members include canine distemper virus, affects dogs, and rinderpest virus. Rinderpest used to be a huge problem for cattle, but it's actually been eradicated now.
Wow.
But what's really unique and significant about measles is that it's primarily a human disease. Yes, primates can get it, but we haven't found any animal reservoir that keeps it going and could reintroduce it to humans.
And that's key for eradication, right? No hidden animal sore.
Exactly. Crucial point.
So how far back does this virus go for humans? When did it become this, like, sustained threat that really stuck around?
Well, it's believed measles only really emerged as a sustained human threat when large cities came into existence. We're talking populations of several 100,000.
Why so large?
Before that, populations were just too spread out. The virus couldn't consistently find enough new susceptible people to infect. These large, dense populations were needed to keep the virus circulating constantly.
Makes sense.
Yeah.
And when did we first actually get a good look at the virus itself, in a lab?
That was a major breakthrough in 1954. Two scientists, Enders and Peebles, they first successfully isolated it from the blood of a patient, David Edmonston.
Edmonston strain, I've heard of that.
That's the one. This isolation was huge because it let scientists finally study the virus up close. It was really the first step towards making a vaccine.
How did they grow it back then?
Initially, they used cells from human or rhesus monkey kidneys,
Yeah.
but over time, challenges with cost, contamination, ethics, it led to using other methods, continuous cell lines, and more recently, specific marmoset lymphocyte lines like B95-8, which are actually better for getting that initial isolation.
Okay, so now we have the virus isolated. Let's pull back the curtain a bit. What did we learn about its actual structure? How is it built? And how does it work inside our bodies?
Right. So the virion, that's the individual virus particle. It's this tiny, kind of irregular, usually spherical package. It's enveloped in a membrane,
How tiny are we talking?
roughly 120 to 250 nanometers across. I mean, you could line up thousands side by side on a human hair. It's minuscule.
Wow.
And inside its genetic material is that single strand of RNA we mentioned, about 16,000 nucleotides long. That's its instruction manual, basically.
And that code's for?
It codes for 6 main structural proteins, including 2 really critical ones on its surface, the hemagglutinin protein, or H, and the fusion protein, or F.
H and F proteins. Okay, so what are they actually doing on the surface? Are they like the virus's keys to get into our cells?
That's a perfect analogy. The H protein is like the grappling hook. It attaches to our host cells.
Got it.
And once it's attached, both H and F work together to fuse the viral envelope with our cell's membrane. That lets the virus kind of slip inside.
So that fusion is critical for infection.
Absolutely. And importantly, if our immune system creates antibodies against either H or F, those antibodies can actually neutralize the virus and protect us. That's key for vaccines.
Okay, so it's inside the cell. How does it replicate? How does it make more of itself?
It does all its replication right there in the cell cytoplasm, the main body of the cell. It completely skips the nucleus for its genetic stuff.
Interesting. Does it shut down our cell's own machinery like some viruses do?
Actually, not really. Unlike a lot of other viruses, measles doesn't significantly interfere with our cell's normal protein-making processes.
Huh. So it's a bit more subtle in that way?
In a way, yes, but it can still cause issues. It can ramp up production of certain stress proteins, transplantation antigens, and sometimes even cause breaks in our chromosomes.
Okay, here's where it gets really interesting for me. We always hear that RNA viruses, like the flu, mutate like crazy, right?
Right, constantly changing.
That's why we need new flu shots every year.
Mm-hmm.
So does measles do the same thing? Is it constantly changing its disguise, making older vaccines potentially useless?
That's a fantastic question, and it's one of the most remarkable things about measles. Despite being an RNA virus, which usually means high mutation rates, measles virus is a single serotype.
Meaning
It's antigenically monotypic. Those key surface proteins, H and F, the ones our immune system targets, they've stayed incredibly stable antigenically over decades.
no way. So they haven't really changed their disguise.
Exactly. Which is just huge for public health. It means the vaccines developed way back from that single Edmundston strain are still protective worldwide against all the wild measles strains circulating now.
That's incredible. A major stroke of luck, it sounds like.
It really is. Makes control and potential eradication much more feasible than for something like flu.
Okay, so if the virus doesn't change its disguise, that's fantastic news for vaccines. But how does our body respond when it sees this unchanging enemy? And what happens if the body can't fight it off effectively?
Well, our immune system throws pretty much everything at it. You get both the immediate, non-specific innate responses and then the targeted adaptive ones.
Innate first.
Yeah. Early on, things like natural killer cells get activated. The body produces interferons, general antiviral signals. Then the adaptive system kicks in. First, IgM antibodies appear, then mostly IgG.
And the antibodies against H and F are key.
Crucially, yes. Antibodies targeting H and F are actually sufficient to protect against infection. But alongside that, you have cellular immunity, your T lymphocytes. That's vital for actually clearing the virus out and recovering.
How do we know the T cells are so important?
We see stark evidence in people with T lymphocyte defects. They often have severe, sometimes fatal measles because they can't clear the virus properly.
Right. And what does measles actually look like clinically? Are there any telltale signs beyond the rash?
So you get that initial prodromal illness, fever, cough, runny nose, red eyes for a few days. But then, sometimes even before the main rash, a really unique sign can appear. Koplik spots.
The Koplik spots.
Yeah, they're these tiny white lesions, almost like grains of salt on a red background, inside the mouth on the cheek lining. They're pretty specific to measles, like an early warning signal.
Huh. Never heard of those.
Then comes that characteristic red blotchy rash. Typically starts on the face, behind the ears, and then spreads downwards over the whole body.
And you mentioned earlier the rash itself is an immune response.
Exactly. It's a consequence of our own cellular immune system fighting the virus in the skin and blood vessels, which is why, paradoxically, someone who is severely immunocompromised might not even develop the rash.
Wow. Okay. What about complications? Are they common?
Unfortunately, yeah, complications are quite common. Up to 40% of cases can have them. The risk is higher in the very young, older adults, people who are malnourished or deficient in vitamin A.
What's the most serious?
Pneumonia is the leading cause of measles-related deaths. Often, it's a secondary bacterial infection setting in after the virus weakens the lungs.
And other things.
Severe diarrhea is common, especially in developing countries. Also, keratoconjunctivitis, an eye infection that can tragically lead to blindness if not treated.
And brain complications. You hear about those sometimes.
Yes, though they're rarer, they're extremely serious. There's post-measles encephalomyelitis, which is actually an autoimmune reaction triggered by the measles infection. And then there are conditions caused by persistent viral infection in the brain, like measles inclusion body encephalitis, MIBE, and subacute sclerosing penencephalitis, SSPE. These are often fatal.
Scary stuff. So how is measles treated? Is there an antiviral drug?
Treatment is largely supportive, managing symptoms, preventing those secondary infections. Vitamin A supplementation is really important. It's been shown to significantly reduce severity and deaths, especially from pneumonia.
Why vitamin A?
Vitamin A is crucial for immune function and the health of epithelial tissues, like the lining of the lungs and gut, which measles affects. Deficiency makes the complications much worse.
Got it. And antibiotics.
Essential for treating those secondary bacterial infections like pneumonia or ear infections. There isn't a specific antiviral drug routinely used, though ribavirin has been tried in some very severe cases, particularly in immunocompromised patients.
So with such potential for severe complications, the vaccine really was a game changer. Tell us about its impact.
Oh, absolutely. It changed everything. Measles vaccines are live attenuated, meaning they use a weakened form of the virus, mostly derived from that original Edmundston strain.
And the impact.
Before widespread vaccination, estimates are around 30 million measles cases every single year globally, resulting in over 1 million deaths annually. The vaccine just drastically reduced that burden, saved countless lives.
Were there bumps in the road? Any early challenges with the vaccines?
Yes, definitely. One big practical challenge is the cold chain. These live vaccines are heat sensitive, so they need constant refrigeration during storage and transport. That's tough in some parts of the world.
Makes sense. Any scientific issues?
Yeah, back in the 60s, there was a formalin inactivated vaccine, FIMV. It was withdrawn because kids who got it sometimes developed atypical measles if they were later exposed to wild virus, a really severe form.
Oof, not good.
Not good at all. Also, some early high-titer vaccines, meant for very young infants, unfortunately led to an unexplained increase in mortality among vaccinated girls in some low-income countries. So those were stopped too. Important lessons learned.
So what about the timing of vaccination now, especially for babies?
Right, that's key because of maternal antibodies. Babies get antibodies from their mothers that protect them initially, but these can interfere with the vaccine taking effect.
So you have to wait.
Generally, yes. Seroconversion rates, the percentage of infants who develop immunity after the vaccine, are lower before 9 months. So the optimal age for the first dose is usually 9 months in high transmission areas, or 12-15 months where the risk is lower. Often given as part of the MMR measles, mumps, rubella vaccine.
And the second dose?
A second dose is often recommended. It helps catch anyone whose first dose didn't fully work, what we call primary vaccine failure. It really boosts population immunity.
Okay, we have to address this. The infamous connection or claimed connection between the MMR vaccine and autism.
Right. It's absolutely critical to address this directly. This concern really took off after a frankly fraudulent report published in 1998, which has long since been retracted.
Retracted completely.
Completely. And since then, numerous huge comprehensive studies, one in Denmark looked at over 500,000 kids, have consistently, unequivocally found no causal link whatsoever between the MMR vaccine and autism. The scientific consensus is crystal clear. It's unfounded.
Good to state that clearly. So given the stable virus, the effective vaccine, is measles eradication actually possible? Can we really wipe it out like smallpox?
Biologically speaking, measles meets many of the criteria. No non-human reservoir, like we said. We can diagnose it accurately. We have a highly effective, long-lasting vaccine.
Sounds promising. So what's the catch?
The catch is its sheer infectivity. Measles is one of the most contagious viruses known to humanity. To actually interrupt transmission everywhere, you need incredibly high levels of population immunity, like upwards of 95% vaccination coverage consistently. That's the main hurdle, achieving and maintaining that level globally.
Okay, so we understand the virus, the disease, the vaccine, but how do scientists actually track it? Especially if the goal is elimination, you need to know exactly where it is and where it's coming from, right?
Exactly. That's where molecular surveillance comes in. It's like viral forensics on a global scale. Absolutely vital.
What does it let you do?
We use it to characterize outbreaks, figure out which strain is causing it, track the transmission pathways, see how it's spreading between people, between regions, even countries, and crucially, provide evidence for the absence of endemic circulation. That's key for verifying elimination.
So it proves a country is truly measles-free, not just having a quiet period.
Precisely. It also helps us gauge how well our surveillance systems are working and measure the real impact of vaccination programs.
So what's the standard tool for this? How do you get the virus's fingerprint?
The standard method for genotyping, giving it that genetic barcode, is sequencing a specific region called N450. It's a 450 nucleotide sequence in the nucleoprotein gene.
Why that specific bit?
It was chosen because it's the most variable coding region in the genome. It changes just enough over time to let us differentiate between different viral lineages or genotypes.
And how many genotypes are there?
Based on N450, the WHO currently recognizes 8 major clades or groups labeled A through H, and within those, 24 specific genotypes. Interestingly, all the vaccine strains are clade A and wild-type clade A viruses. They're now considered extinct, wiped out by vaccination.
Wow, that's progress. So where does all the sequence data go? Is there a central database?
Yep. It all gets submitted to the Measles Nucleotide Surveillance Database, or MEANS. It was set up back in 2005 by Public Health England and the WHO.
And each sequence gets a name.
Right, a standard WHO name. It includes info like where and when the case was found, something like MVS-Georgia.USA 10.24, meaning a measles virus from Georgia, USA, case number 10, identified in 2024, allows for global tracking and comparison.
Okay, this is fascinating. If N450 is the standard, why isn't it always enough? You hinted that our success is actually creating a new challenge.
You got it. That's a really important point. Because global vaccination has been so successful, we've drastically reduced the number of circulating wild-type genotypes.
Fewer strains around.
Exactly. For instance, since 2021, really only 2 genotypes, B3 and D8, have been consistently detected globally. Genotype H1, which used to be common in China and often exported, hasn't been seen globally since late 2019, thanks to their vaccination efforts.
So the problem is, if everyone has B3 or D8,
the diversity has shrunk so much that N450 alone often doesn't have enough resolution to distinguish separate transmission chains, especially when different outbreaks are caused by genetically identical viruses based on that N450 sequence.
So what are scientists doing to track within these dominant B3 and D8 genotypes? How do you get more detail?
Great question. The Global Measles and Rubella Lab Network, GMRLN, developed new tools. First, distinct sequence identifiers, DSIds.
DSIds.
Yeah. Every new unique N450 sequence submitted to MEANS gets its own unique DSId code. Any later submissions of the exact same sequence get that same DSId. So it flags identical viruses found in different places.
Exactly. It lets scientists quickly spot identical sequences popping up, say, in different countries, which strongly supports epidemiological data about whether a case was imported or not. We see different DSIds are more common in certain parts of the world.
Okay, that helps. What else?
Second, there are named strains. These are specific DSIds that have become really common, submitted at least 50 times from multiple countries over 2 years. They represent variants that are clearly circulating widely globally.
So finding a named strain tells you something specific.
It tells you that the virus is part of a well-established, globally circulating lineage. The name comes from the WHO name of the first submission. But importantly, that doesn't mean that's where an importation came from now, because these strains are everywhere.
Got it. So DSIds and named strains add detail to N450. But it sounds like sometimes you need even more resolution, like for really fine-grained tracking. You're absolutely right. While N450 is still the standard for initial genotyping, its resolution is limited for tracing specific transmission pathways, especially when you're close to elimination and every link matters. So we need extended sequencing approaches.
Like what?
One approach is sequencing the matrix fusion non-coding region, or MFNCR. It's about 1,000 nucleotides long, located between 2 genes. It's one of the most variable bits of the whole genome.
More variable than N450.
Significantly, so it gives you better phylogenetic resolution, a more detailed family tree of the virus. Plus, it can be sequenced using standard lab methods, Sanger sequencing, which makes it more accessible for many labs.
Okay, and the ultimate approach?
That would be whole-genome sequencing, or WGS, sequencing the entire 16,000 nucleotides. That gives you the most complete picture of the virus's evolution and relatedness.
Where is WGS being used?
It's currently done in fewer, more specialized labs using advanced next-generation sequencing, or NGS platforms, but it's been incredibly powerful for dissecting complex outbreaks. For example, it helped untangle measles transmission among Afghan evacuees resettled in the U.S. in 2021, linking cases that weren't obviously connected epidemiologically.
Powerful tools.
Yeah.
But you mentioned accessibility. Are there challenges getting these advanced methods like WGS used everywhere?
Absolutely. WGS requires serious computational power, specialized software, and bioinformatics expertise to analyze the massive amounts of data. That's not readily available in all national labs, particularly in lower resource settings.
And just getting the right samples can be an issue.
That too. Despite recommendations, there are still big gaps in molecular surveillance, especially where measles is still endemic. Often it's because collecting the right kind of sample, like a throat swab or urine needed for sequencing, isn't prioritized. Serology, just checking for antibodies in blood, is still often the main confirmation method.
So even with the tech, logistics, and infrastructure matter.
Hugely. But it's also worth remembering this molecular data has other uses too. It helps us constantly check that our diagnostic tests are still working, making sure the virus hasn't mutated where our tests bind.
Ah, quality control for the tests.
Right. And critically, it provides the data to monitor viral evolution against vaccine efficacy, making sure our current vaccines are still holding up and giving us the info we'd need if, heaven forbid, an updated vaccine was ever required.
Right. Continuous vigilance.
Exactly.
So what does this all mean for you, the learner? Well, it means that behind this goal of eliminating measles, which sounds simple enough, there's this incredibly sophisticated, interconnected, global scientific effort going on.
Yeah, from understanding tiny viral proteins to tracking subtle genetic changes across continents, molecular epidemiology really gives us the eyes to see these invisible pathways of disease.
It feels like a constant race, doesn't it? Scientists trying to anticipate how the virus might evolve, adapting their tools to stay one step ahead.
It really is.
And this deep dive, I think, highlights that getting to a truly measles-free world isn't just about hitting vaccination targets. It's also about this continuous, rigorous surveillance, the tireless work in labs worldwide using these cutting-edge molecular tools.
Absolutely. The lab network is the backbone of knowing where we stand.
It really makes you wonder, though, as global travel gets even more complex and as our vaccines keep pushing the virus evolutionarily, what new molecular challenges might pop up? And how will this scientific detective work have to adapt again to track and ultimately, hopefully, defeat this incredibly persistent virus? Okay, let's unpack this. Measles. You know that childhood illness we mostly hear about as a thing of the past? Or maybe a